cell death essential for immune system function. Apoptosis can be initiated through two main pathways, intrinsic and extrinsic. In the intrinsic pathway, cellular stress causes mitochondrial outer membrane permeabilization. This releases cytochrome C into the cytoplasm, which forms the apoptosome complex with APAF1 and pro-caspase 9. The activated caspase 9 then activates executioner caspases. In the extrinsic pathway, death receptors on the cell surface bind to their ligands. This binding recruits adapter proteins that form the death-inducing signaling complex, or DISC. The DISC activates initiator caspases, like caspase 8. Caspase 8 then activates the same executioner caspases as the intrinsic pathway. These executioner caspases cleave cellular proteins, leading to characteristic morphological changes. The first observable change is cell shrinkage. Next, the chromatin condenses and DNA fragmentation occurs within the nucleus. The cell membrane begins to bleb, forming bulges on the surface. Finally, the cell breaks apart into apoptotic bodies, which are phagocytosed by immune cells without triggering inflammation. Apoptosis is crucial for immune system function, helping maintain cellular homeostasis and preventing autoimmunity. The immune system utilizes several specialized types of apoptosis to maintain proper function and homeostasis. Activation-induced cell death, or AICD, eliminates activated T cells after they've completed their immune response. This process is crucial for preventing autoimmunity and maintaining the proper balance of lymphocytes in the body. Negative selection in the thymus is a process that eliminates developing T cells that could potentially attack the body's own tissues. This elimination of self-reactive T cells through apoptosis is a critical process for establishing central tolerance and preventing autoimmune disease. Cytotoxic T lymphocytes and natural killer cells are specialized immune cells that can induce apoptosis in virus-infected or tumor cells. They use two main pathways: release of perforin and granzymes that create pores in the target cell membrane, or through Fas ligand interaction with Fas receptors. Both mechanisms activate the caspase cascade in the target cell, leading to its controlled elimination through apoptosis. Neutrophils are the most abundant white blood cells and are often the first responders to sites of infection or inflammation. These short-lived cells undergo apoptosis after they've performed their function, which is crucial for resolving inflammation. The timely removal of neutrophils through apoptosis helps limit tissue damage and is essential for the proper resolution of the inflammatory response. Dysregulation of these apoptotic processes can lead to various immune-related diseases. Insufficient apoptosis of self-reactive immune cells can result in autoimmune diseases where the immune system attacks the body's own tissues. Excessive apoptosis of immune cells can cause immunodeficiency, reducing the body's ability to fight infections. When apoptosis fails in abnormal cells, it can contribute to cancer development, allowing malignant cells to continue proliferating. The balance between pro-apoptotic and anti-apoptotic proteins regulates apoptosis in the immune system. Pro-apoptotic proteins like BAX and BAD promote cell death, while anti-apoptotic proteins like BCL2 and BCL-XL inhibit it. This delicate balance makes these proteins potential targets for immunotherapeutic interventions in various diseases.